Good morning, everyone. So, in the last class, we discussed about archaebacteria and archaebacteria's different kind of archaebacterias that are present. We talked about methanogens, which are present in the rumen of the you can say herbivorous animals such as cows and buffaloes. Then we talked about halophiles, which occur in salt-rich areas such as marshes. Example of halobacteria, halobacterium and halococcus. Then we talked about thermoacidophiles, which are able to tolerate high temperature as as well as high acidity or the low pH. They can survive in the hot sulfur springs where the temperature can reach up to 80 degrees Celsius with the pH as low as 2. So then we talked about some more general points about the archaebacteria, that gram staining is not usually referred or utilized for archaebacteria classification because they do not have peptidoglycan in their cell wall. So we later on discussed a bit about gram staining. In today's class of the U bacteria, we are going to discuss more about the gram staining in detail. First, let's talk about U bacteria. Bacteria was discovered by Antony von Leeuwenhoek. Antony von Leeuwenhoek is also called as father of microbiology, and there are a lot of stories about how he actually worked with the you can say a lens which he utilized as a you can say a microscope so as to study a lot of different small organism that we call as microorganisms. He discovered bacteria in 1676. Then we have some general characteristics of the you can say bacteria. Bacteria are unicellular forms, and bacteria have peptidoglycan in their cell wall. If the cell wall is thick, it is gram positive. If the cell wall is thin, it is gram negative. We'll go into much more detail later on. The gram positive have teichoic acid, one of the components of the cell wall, which is present in gram positive, and teichoic acid is absent in gram negative. We'll talk a bit about gram bacterial plasma membrane. Bacterial plasma membrane, gram positive bacteria have phospholipid bilayer, but gram negative have inner cell membrane, which is made up of phospholipid. So we'll go into the much more detail about the structure later on. These are the general characteristic. Mucilage covering is present and naked circular DNA is present in the form of nucleoid. We'll go into details about all this later on. Membrane covered organelles such as endoplasmic reticulum are absent. That's why they are called as prokaryotes, because they do not have membrane bound organelles. Ribosomes are 70s in nature. Ribosomes are non membranous cell organelle, which means this is an organelle which does not have a cell, you can say a membrane outside. That's why they are present in prokaryotes, as they are non-membranous. We'll go into the detail about what this 50s plus 30s for stands for in the later section of this class. Genetic material is in free naked state and also called as prochromosome or nucleoid. Size of bacteria varies from 2 mm to 2.6 micrometer. When we are talking about this in meter, it is 2 into 10 power 3 meter to 2.6 into 10 power minus 6 meter. So this is the range of the size of bacteria. One of the smallest bacteria that we know is Dialister pneumocyntis. The size of it is 0.15 micrometer. We also talk about mycoplasma. Mycoplasma is also very small bacteria. Largest bacteria category belongs to spirochetes and blue green algae, where the size is 500 micrometer. So then we come on shapes of bacteria. What are the different shapes of bacteria? We can classify bacteria on the basis of the shapes that they are present in. First shape is coccus. Coccus shape means they are spherical or ovoid in outline. Depending upon the group, if they are one, then they are monococcus. If they are two, then they are diplococcus. If they are in a chain like this, then they are called as streptococcus. If they are in a cluster, then they are called as staphylococcus. One of the example of coccus bacteria is staphylococcus aureus. This bacteria is present on the skin of human beings, on mostly areas like face. When the you can say the population of staphylococcus aureus grows on our face, it can lead to problems such as acne or pimples. Then we have second shape, which is bacillus. Bacillus are small rod-like or cylindrical shaped bacteria. It can be diplobacillus or streptobacillus. Streptobacillus as in a chain of bacilli. One of the example of bacillus is lactobacillus. Then we have spirillum, which is spiral shaped and coiled like a screw. Example of it is spirillum, spirochetes. Aggregation does not occur. So when we are saying about, you can say this, a chain or a cluster, aggregation does not occur in spirillum. Then we have vibrio. Vibrio is comma shaped. So if we are talking about the shape, this is somewhat the shape of vibrio. Example of it is vibrio cholera. Vibrio cholera causes cholera disease in humans, and it lives alone. You do not see, you can say vibrio or staphylovibrio, or you can say streptovibrio. It is singular that we see in nature. Then we talk about flagellation. When we are talking about, we'll go into the structure later on. But the structure of flagella is, we go into that detail later on. But when we are talking about flagellation, the flagella, if it is single in nature, if it is only one, then we call it as monotrichous. Single flagella occurs at the near of the end of bacteria. One flagella, this is monotrichous. Amphitrichous, a flagella at each end of the bacterium, that is amphitrichous. Cephalotrichous, a cluft or a group of flagella at one end. Lophotrichous, a group or cluft of flagella at both ends. And then we have peritrichous. Number of flagella are distributed all over the surface of the bacteria. This is one of another category of way to classify bacteria on the basis of flagellation. Then we have structure of bacteria. What is the structure of bacteria that we see and we study? I've been talking about gram staining for a long time. Gram staining process, first we have to see the process. We take a, let's say, bacterial smear. How we exactly create a bacterial smear? Let's say we have a patient at hospital that has blood infection with one of the bacteria. Let's say staphylococcus aureus is present inside the blood, and then it is causing infections to the patient. Then we take the blood sample, and then we put few drops of blood sample on a, you can say, glass slide, and we create a bacterial smear. Take this glass slide and we put some drops and we smudge it. We make a rounded smudge of it. Then we heat fix it. We pass that, you can say, glass slide over the flames, and we heat fix it so that it dries up. Then we add crystal violet on the glass slide, add some iodine, which is also called as mordant in this case. So iodine fixes all the crystal violet and with the bacterial smear. Then we decolorize it. This crystal violet is purple or violet in color. Then when we decolorize it, all the color goes away in the case of gram negative, but the color stays in the case of gram positive. Then we add counter stain, which is saffronin, which is red in color. So what I am going to explain is, when we have a, I have already told, the cell wall of gram positive is thick and the cell wall of gram negative is thin. So when we add crystal violet, the crystal violet deeply is absorbed in the gram positive cell wall, and it is not very deeply, you can say, absorbed in the gram negative cell wall. So when we remove the color through, you can say, ethyl alcohol, or acetone plus ethyl alcohol, then the color is not easily removed from the gram positive, but it is removed from the gram negative as it is a thin cell wall. So then we, after that, we add safranin, which is the counter stain. It is already, you can say, white in color after the crystal violet has been removed in the gram negative, but the crystal violet is present in the gram positive. So gram negative absorb the red color, and gram positive does not absorb the red color as it already has the violet color. So we can say the we can say the gram positive will appear purple in color and the gram negative will appear pink or red in color because of the presence of, you can say, a safranin, which is a counter stained it. So now we see the structure of gram positive and the gram negative bacteria. What are the other components of the gram positive and gram negative bacteria apart from the cell wall? So there is a mucilaginous sheath that we just talked about earlier. There is a mucilaginous coating above the, you can say, bacteria, which is present in both gram positive and gram negative bacteria. Then there is a coating called glycocalyx. If the glycocalyx is tough, then or you can say solid in nature, then we call it as capsule. If the glycocalyx is loose in structure, then we call it as slime layer. Then we have cell wall. In case of gram staining, when we are doing it in the gram positive, the cell wall looks purple in color. And as we have seen, there is a thin peptidoglycan in the cell wall of the gram negative, which appears pink and red when we do the gram staining. But apart from the thin peptidoglycan, there is an outer membrane of cell wall which is present in gram negative. We also we'll study more about this later on, what exactly is the outer membrane of cell wall. Then we have plasma membrane. It is, plasma membrane is thicker in gram positive and thinner in gram negative. Then we have the cytoplasm, which is common. So what exactly are the changes that we are seeing? Gram positive, this mucilaginous sheath is same, this is same, the glycocalyx bead capsule or bead slime layer, it can happen in both of the cases, gram positive or gram negative. Then cell wall is different, as cell wall is in one component which is in gram positive and two components in, you can say, gram negative. One is outer membrane of cell wall, and then there is thin peptidoglycan cell wall. Then we have a thicker plasma membrane in gram positive. Then we have thinner plasma membrane in gram negative. Then we have the common cytoplasm, which is similar. So when we are talking about different kind of features that we see in gram positive and negative, gram positive are usually less pathogenic. They are, you can say, they infect, the chances of infection from the gram positive are lesser, and you can say the gram negative are usually more pathogenic. Teichoic acid again is present in the gram positive bacteria and absent in the negative one. We talked about what exactly is the process of conducting a gram staining on a bacteria. Then here I have some one of the picture that I'll show it to you for a general photo of, or a general structure of gram, not just gram, but a general photo of a bacteria. So here you see one of the structure inside is nuclear body that we called as nucleoid. We also called it as prochromosome. Then we have the plasma membrane inside. Inside plasma membrane is there. Then outside we have cell wall. This is the cell wall. And after that there is a glycocalyx. It can be a capsule or a slime layer depending upon the composition. Then outside this is also a mucilaginous covering. Here we have a, you can say, a flagella. Flagella components are there. This is the hook part. This is the filament part. We'll go back into the details about the components of flagella later on. So, and this is the lipid globules. Lipid is present inside, the fat is present in the form of, you can say, globules, which is scattered inside the cytoplasm. And these small structure, we will go into the later, is it a pili or is it a fimbriae? We'll go into the details later on. Then we have plasma membrane. When we are talking about structure, plasma membrane is selectively permeable. What exactly do we mean when we say it is selectively permeable? It allows entry of only selected material. Let's say there is a bacteria. This is a, you can say, flagella, cell wall, and the cell membrane. Once the material has crossed the cell wall, to allow the material to go inside the cell, or you can say inside the cytoplasm, cell membrane has to give the permission to that material. It is selectively permeable. If it wants to allow that material to come inside, it will allow. If it does not want to, it will not allow. Multiple layers of proteins are present with phospholipid. When we are talking about the component of plasma membrane, as we talked about, it is phospholipid in nature. So, along with phospholipid, there are type of proteins we'll talk about later on. What are the proteins? They can be integral or transmembrane. We will talk about later on. These proteins are present along with the phospholipid. The plasma membrane is metabolically active for respiration and synthesis of lipid and cell wall components. So, what exactly do we mean when we say they are metabolically active? I mean, when we have, you can say, a eukaryotic organism, let's say a plant cell that we have. Plant cell is doing the respiration and the generation of energy through, you can say, different kind of cell organelles which are membranous in nature. That are, you can say, chloroplast is there, which is helping in generation of energy. Then we have mitochondria. These are all organelles that have membrane. But in case of a prokaryote like bacteria does not have mitochondria because it is prokaryotes are membrane less membrane non-membranous organelles which are present inside the bacteria. So mitochondria is not present in prokaryotes. Endoplasmic reticulum is not present, and chloroplast is not present. So instead, they have a different kind of processes that are going inside the, you can say, you can say inside the plasma membrane only. So this is the, let's say, a bacteria, and bacteria's plasma membrane is this. There is a organelles, or you can say structure called mesosomes. Mesosomes, they have enzymes fitted inside them which helps them in respiration. Instead of mitochondria, mesosome is doing the work inside. So metabolically active for respiration, and they also synthesize lipids and cell wall components. So cytoplasmic streaming is absent in prokaryotes. What exactly is cytoplasmic streaming? Cytoplasmic streaming helps in rapid movement of substances in cytoplasm. When we have a bacteria, the cytoplasm inside it is not moving. It is stationary. But in eukaryotes, we see cytoplasmic streaming. The material keeps on moving and mixing, which helps in rapid movement of substances in eukaryotes, but in prokaryotes, it is absent. We have sap vacuoles which are absent, but we have gas vacuoles which are present. There are cytoplasmic structures which is inside cytoplasm, or you can say made up of plasma membrane. Mesosome, as we already talked about, this is, let's say, a mesosome. This is a mesosome that is present. Mesosome are villiform structure. You can say villi made. These are finger-like projections that are present and developed from plasma membrane. Mesosome are of two types. One of the type is septal mesosome. It connects nucleoid with plasma membrane and takes part in replication of nucleoid for providing points for attachment, helps in septum formation. What exactly do we mean by this? When we are saying you can say a bacteria have to divide. Then the nucleoid is present inside, and for nucleoid to divide in half when the bacteria is going through binary fission, the mesosome is going to grab the nucleoid from the one side. One mesosome is going to, you can say, connect with the other part of the nucleoid, and it is going to pull the half of the nucleotide here, half to the nucleoid here. Then nucleoid is going to get split from the half, and then we are going to see two different daughter cells. So this is what the septal mesosome does. Then we have lateral mesosome. As we talked about, lateral mesosome, a bacteria does not have chloroplast, or you can say mitochondria. So there are lateral mesosome present. Lateral mesosome have enzymes present, and that enzymes is going to do the respiration, as it possesses the respiratory enzyme. And the whole you can say lateral mesosome is also called as chondroid. Then we are going to talk about ribosomes. Ribosomes are small membrane-less ribonucleoprotein. So you can say the components of ribosomes are you can say RNA is present, plus some proteins are present. So ribonucleoprotein, they are of two types. Ribosomes can be of two types: free fixed ribosome that are attached to the plasma membrane, and then there we have free ribosome which are freely moving in the cytoplasmic matrix. Role of ribosome is to make proteins. How exactly do they make proteins? We'll study later on. You most probably you'll study this about in 12th standard, when we study in molecular basis of inheritance, and the translation you'll study. In translation topic, you will study about how exactly do we make proteins from a ribosome. When ribosomes occur in a helical group, they are called as polyribosome or polysomes. Now we are going to talk about nucleoid. Nucleoid represents the genetic material of prokaryote. We do not have a nucleus inside a you can say a prokaryotic cell or a bacteria. This nucleoid is also called as genophore, prochromosome, and chromoneme. A bacterial cell can have two or more nucleoids, but all are replicated copies of the same nucleoid. When we have, say, there is a uh, multiple nucleoids present inside a bacteria. So what we mean by that statement, it is that all three, there can be more than two present nucleoid present, but all of them have arisen from one single nucleoid. Nucleoid is attached to plasma membrane through mesosome. Which exact mesosome? Septal mesosome. So this was the first part of the general characteristics of the bacteria. We'll study more about the general characteristics in the next class. Then we'll study about you can say respiration. Then we'll study about reproduction. So stay tuned with this series, and I hope that these lectures are helping you out. Thank you so much.